Hi guys, welcome to another chemical engineering tutorial brought to you by the ChemEng student. In this lesson we're going to take a look at the complete design of a bioreactor and all the key parameters associated with microbial growth and how they influence bioreactor design. So the first thing that we'll take a look at is the definition of a bioreactor. And a bioreactor can be described as a vessel or a container in which biological reactions are carried out. And this is especially true for microorganisms or cells. Now, these vessels are employed in the production of several biochemical compounds and often involve the cultivation of biological species. Now, some of the things that we would like to produce in bioreactors are things like antibiotics, vitamins, even alcohol, and insulin, just to name a few. Now, the bioreactors can sometimes also be known as fermenters, and they typically do take the form of batch processes, because ultimately they will have a certain period of time that they need to take before their growth can be realised. And we'll see this in just a second in the growth curves. But we've got several factors that are associated with the design and indeed the operation of bioreactors. We've got things like the agitation and the mixing. We've got the temperatures, the light, the pH and the gas exchange. Now, this is something that I do as part of my research, is I look at high value biochemical compounds from a wide range of microalgae species, and the design of reactors and the manipulation of these variables is something that I do quite frequently. So certainly in the context of bioreactor design, these things, especially the agitation and the temperature, are very, very important. Now, it depends on the type of species as to whether or not we can design what's called a photobioreactor. And this would be the inclusion of lights. Some systems do not require high amounts of light. Other systems do require considerable amounts of light. So it's just worth keeping that in mind. Now, generally there are three types of bioreactors in the context of either lab scale or industrial scale. Now, variations of these do exist. Some are highly specialised for a specific species, others are very generic, but they usually take the form of one of these three systems, whereby we have a stirred tank reactor, we have an air-lift reactor in which we basically have this centre portion whereby we inject a gas, usually in the form of air, but it can be carbon dioxide enriched air. And then what happens is we overspill the bubbles, and this induces some mixing as well. Now, the more common one, or certainly for me, is the use of bubble column. Now, the bubble column acts in a similar way to the stirred tank. The only thing that we don't have is the stirrer within the system, because what we do is this sparger at the bottom, which contains small holes, we allow aeration to perform the mixing function. Rather than have mechanical mixing, we have gaseous mixing. Now, when it comes to the mass transfer within bioreactors, we have two key components that must be addressed. Those are the oxygen and the carbon dioxide transfer. And which one is desirable and undesirable will depend upon the type of biological species being cultivated. Because ultimately, some will require oxygen to respire, other ones will require carbon dioxide to respire. Now, in order to work out which one is which, we have two types. We have aerobic cultures. These ones require oxygen. Now, this is just a very generic example there is a whole host, there is hundreds of thousands of different species of bacteria, microalgae, macroalgae. So this is just a very generic image to show you an example of these microbes. But we have aerobic cultures. These are ones that require oxygen and will expel carbon dioxide. We have anaerobic cultures which require carbon dioxide and will expel oxygen. Now, usually in anaerobic cultures, these are more associated with plant-like behaviour because they absorb UV light and they will consume carbon dioxide in order to photosynthesize. Whereas aerobic cultures like bacteria need the oxygen in order to respire and produce carbon dioxide. Now, before we start to look at the design of a photobioreactor, it's really important that we understand the behaviour and the growth of microalgae from a very general standpoint. And we can basically break up the growth of microalgae into 4 distinct stages. We have a lag stage, we have the exponential stage, a stationary stage, and a death stage. Now, the lag phase occurs at the very beginning of the cultivation. So, for example, if in my research I am going to cultivate some new microalgae for experiments, then as soon as I put in the seed culture, which we sometimes refer to this as the inoculum, then when we put the inoculum in the medium, this will be the lag phase. So what this implies is that there is no real growth at this moment in time, and this can usually last a couple of days, depending on the species. But this is basically the time in which the microalgae or bacteria or macroalgae are adjusting to their new surroundings. The exponential phase, or the log exponential phase, is where the most amount of growth will take place. So it's usually quite rapid in their multiplications. So, like 1 makes 2, 2 makes 4, 4 makes, 16, and so forth. So you can see that the growth becomes exponential. And this doesn't usually last too long. It's usually done within, certainly for me, within about a week, depending on the species. Now, the stationary phase occurs when no new cells are created, but rather the cells begin to mature and start to produce certain high value compounds. And then after a certain amount of time, then the population will start to decline because the cells will begin to die off. And this is usually due to a lack of nutrients, because within the growth medium, they have consumed the nutrients and therefore they have no longer got a solid food supply. Now, within each one of these phases, probably neglecting the lag phase, the microalgae or macroalgae will produce different types of compounds. So if we want to exploit through our photobioreactors or our bioreactor designs, if we want to exploit certain components, we need to fully understand the growth curve associated with our species. Now, the bioreactor design equations are fairly simplistic, but can sometimes be a bit abstract to get used to in the context of standard reactor design. Because, for example, if we want to think of what is the main objective of the system, then the things that we are interested in is the batch time, the specific growth rate of the algae, the substrate concentrations, and the biomass productivity. Granted, there are more, but these are the most fundamental elements of any bioreactor design. Now, this, just to give you some context, is a photo of a microalgae species that I actually work with called Botryococcus branii. And you can see here that this is a colonial microalgae in that it is bound together by oil. So that's one of the things that I'm involved in, is the extraction of this particular oil. Now let's take a look at all of the equations that we're going to use before we apply it to an example. So the first one is the biomass. So the biomass is given by capital X, and this is going to be equal to the final biomass, so that will be XF, minus the initial biomass multiplied by the working volume of the reactor. Now the total production time, or the T with a capital T, is equal to the batch time, so that's TB, plus the downtime, which is TD. Now the downtime can be associated with the removal of the old material, the sterilization, and then the addition of the new material. So that is the time in which the reactor is not working. Now the number of batches will be the time divided by the total time. And that will tell us that depending on how many days we want to operate, how many hours we want to operate, if we divide this by the production time, then we can work out how many batches we will have within a given period of time. Now the annual biomass, denoted here by X prod, is simply going to be the biomass value multiplied by the number of batches, because this is the biomass per batch. So if we multiply these two together, then we can get our annual biomass productivity. Now we have an exponential growth which gives us the relationship between biomass and time. And it is an exponential factor because we've seen that in the growth curve. And that is equal to the initial biomass concentration, e to the power, and this is mu max multiplied by t. Now mu max is the maximum growth rate. So that is the quickest growth rate that is associated with this cultivation, i.e. how quick are these cells multiplying. We then come to the substrate concentration, and the substrate can vary depending on the system that you're using. It could be a specific compound, it could be a specific salt within the culture medium, it could be a wide range of things. But here we will denote the substrate concentration for the initial amount as either Si or S0. The final substrate concentration we can denote as SF, or sometimes known as S. It just depends. Now the biomass yield given by Y, and this implies here, this is the yield of the biomass with respect to the substrate. I'm going to just summarize this and just give it Y so that it's a bit tidier when we are performing the equations, but usually you would denote the subscript of X over Y to tell us that it is the ratio of biomass to substrate. So the change in biomass against the change in substrate. And then the batch time is given by TB is equal to 1 over the maximum growth rate, the log of 1 plus the biomass yield divided by the initial concentration multiplied by the change in the substrate concentration. Now this equation is really important, and it's based on this idea of the reaction kinetics. And the reaction kinetics, as we know from normal reactor design, is highly influential on all aspects of the reactor design. And so this is one of the things that should be fundamental in your design, and one of the first things that you consider. Now here what we're going to do is we are going to consider the use of the Monod equation. And this basically takes a similar form to the Michaelis-Menten equation, which the key distinction here is that this one is an empirical nature and is based on a lot of theoretical considerations. But ultimately this is the kinetic equation that we're going to use. And this is our growth rate is equal to our maximum growth rate multiplied by the substrate concentration divided by the rate of substrate consumption plus the substrate concentration again. Now, the proof of the batch time is going to be taking place through the following sets of assumptions. We want to simplify the rate, so for this particular rate expression, this will apply when the Ks is significantly smaller than the substrate concentration, which in the example that we're going to look at, it is certainly a lot smaller. Now our growth rate, we're going to say that mu tends to mu max. And from here, what we can then do is we can simplify the Monod equation in order to express mu in terms of mu max and the substrate concentration explicitly. So from here, we can now bring in our biomass yield with respect to the substrate, and we can rearrange this for x. Because ultimately we need to express x as a function of substrate and yield. So just by simple algebraic manipulation, we can express x in this form. Now if we bring in the exponential equation, then what we can also do is re-express this for batch time, because this is the only place in which time appears. So in all the other equations, we haven't seen the batch time. Now granted, this t could be any specified time, but because we're talking about the context of batch, we'll give it its subscript b. Now simplifying this, we take the logs of each side, we'll divide by mu max, so we see here that we get 1 over the maximum growth rate log x over x naught. Now we're almost there. We can now substitute in x into this final equation, and what we end up with is the equation for our particular batch time. So you can see the derivation, and you would basically apply this to any other given system that you want. Now some other models for you to consider are some given by this figure here, and this figure can be found by using this DOI. So this is the unique identification code of that particular publication. I'll put a link in the description to that publication if you want to go and check that out. Now let's take a look at a working example here. So we have a 1,000 litre stir bioreactor which uses methanol to produce a single cell protein used in the treatment of certain infections. The biomass yield from the substrate is 0.52 gram per gram, with a substrate affinity constant, so that's the rate, of 0.7 milligrams per litre, and a maximum growth rate of 0.55 per hour. So that's our mu max. Now the growth rate contains 4% weight per volume of methanol, and a desired substrate conversion of 98% is basically desired, and the initial cell concentration is 0.1 gram per litre, with a downtime between each batch of 5 hours. We want to determine what the annual biomass productivity of this particular system will be. So quite a few steps to go through, but as you'll see, every equation that we've looked at, we will need to apply it. Because ultimately, the formula that we need is going to be our biomass multiplied by the number of batches. Now, we can expand on the dependencies of these two parameters, because we now know that the batch time is the overall time divided by the total productivity time. We know that x is the difference between the final and initial biomass concentrations multiplied by the volume, and we know that the total time is equal to the batch time plus the downtime. So now we can see that ultimately, in order for us to solve this, we need to find the batch time, and we do that using this equation. Now the thing that we don't have though is the S values explicitly. We need to express these in the initial and final substrate concentrations of mass per unit volume. Now we know that there is a 98% conversion and a 4% weight per volume initial substrate concentration. So what we can actually do is express S naught, which is the initial, so we can convert this percentage and get it into 40 grams per litre. Now from here, if that's the initial amount, the final must be the 2% that is unreacted multiplied by 40, which gives us 0.8 grams per litre. So we now have all the values to substitute in to the batch time. If we solve this, we get an approximate batch time of 9.676 hours. Now we can determine the total time by simply adding our 9.676 plus our downtime, which was 5 hours. So we get a total time of 14.676 hours. Now the batch time can be found, the number of batches can be found, whereby we take the total duration divided by the total time. Now we want to operate 365 days and 24 hours a day. Yes, this is unrealistic, but this is what we are just going to assume in this very hypothetical, fictitious example. But ultimately, this wouldn't necessarily be the case unless we had multiple reactors running in parallel. But after performing this calculation, we work out that within the amount of time in that year, we'll go through 597 batches. So now the final thing that we need before we work out the annual biomass is the final biomass concentration. And we do this using the batch time exponential equation. So we have our mu max, we have our batch time, we have our initial concentration, so we can get a final concentration of 15.3 grams per litre. So now we can determine the total mass as a function of the working volume, which will give us 15,200 grams. Now therefore the annual biomass can be deduced as 15,200 multiplied by the number of batches, which will give us 9074400 grams per year, or in other words 9.07 tonnes per year. So you can see that the equations are nice and straightforward, but they have to be solved in a very systematic method. Now just some supplementary material, because this is my field of expertise. If anybody's interested in seeing more about my research, about my work, would like to get in contact with me, I always work with the media where possible. I like to collaborate, work with universities, with companies. So you can see here, I'll put all these links in the description, but you have my ResearchGate profile. I have an article in the Independent newspaper. I also have articles in The Conversation, and these are just some of the associations that I have between the University of West of Scotland, where I currently work, the University of Glasgow, Strathclyde, the University of Tokyo, and so forth. If you also want to connect with me on LinkedIn, then you can scan this QR code. Feel free to connect and send me a message if you're interested in learning more about my work, my research, you'd like to collaborate, you'd like some advice, etc. So please feel free to get in touch. So that's the end of this lesson. Thanks for watching. Hopefully this was helpful in understanding the concept of microalgae and macroalgae growth, also bacterial growth, and how that relates to the design of bioreactors, the different types of bioreactors, and the different types of kinetic models. If you like this video, please like and subscribe to the channel. It really helps us reach as many students as possible. Thank you for your time, and look forward to seeing you in another video.